In this video, we're going to do the second half of how DNA becomes a protein. The other day, we talked about step 1, transcription. DNA becomes RNA. In step 2, translation, the step we're going to talk about today, RNA becomes proteins. This process doesn't have too many steps, similarly to transcription. Translation doesn't have many steps, but the steps are a little more complicated and harder to understand. So make sure you rewatch parts if necessary. Let's start out by saying what is translation. The word translation has many meanings, but in science, translation is the process of RNA becoming a chain of proteins. Proteins are what your body actually uses to make you look a certain way. They're the code your body can use. Your body doesn't directly use DNA. Therefore, it needs to take DNA, turn it into RNA, which we learned about the other day, called transcription, and then translation is the process of RNA becoming chains of proteins. Before we can talk too much about what translation is, there's a couple of vocab words you need to know. The first vocab word is a codon. A codon is a group of 3 nucleotides. And if we think back, nucleotides are the building blocks of DNA and RNA. Therefore, if you take 3 nucleotides, like A, U, and C, this is a codon, the grouping of A, U, and C together. Another codon would be GAU, or the next 3 nucleotides in the RNA sequence given in the example. And finally, the last codon would be CGG, or the last 3 nucleotides in the RNA sequence. Codons are the language your ribosomes read, so they think of them like 3-letter words. Your ribosomes don't read or look at your RNA as AUC, but they see AUC as a single word. So what happens in translation is your RNA is read by codons. So your ribosomes are going to read your RNA as codons. Before we talk about all the steps of translation, try this practice problem. How could you break up this sequence into codons? Look at the sequence and write down or circle maybe the first 3 codons just for practice. If you've done that, what you should see is this would be 1 codon, the second codon would be CUA, GCU, GGG, etc. Quick review before we move on with translation. Where do proteins get made? What organelle? And what is the monomer of proteins? Write these answers in your notes. So if you remember that proteins get made on ribosomes, and the process of translation, the purpose of it is to make proteins, then you could figure out that translation also takes place on ribosomes. What happens in translation? The first step of translation is mRNA, which was transcribed in the nucleus, travels and attaches on to ribosomes, either attached to the ER or free ribosomes. Step 2 is a molecule called tRNA, brings amino acids to the ribosomes, and the tRNA molecules match up with the codons, or 3 letters, words, on the mRNA and form amino acid chains. So essentially what happens is you have your mRNA strand here, it attaches onto your ribosome, and tRNA, which is represented by these little yellow circles, read the words like AUG and say, oh, AUG codes for or is the amino acid methionine, which is attached to the top of the tRNA strand. So it attaches a methionine onto the original mRNA. This keeps happening to the whole mRNA strand. So for instance, we still have the same mRNA right here, still attached on to the ribosome, but now the tRNA is looking at the next part of the sequence, so GCC codon. It says, oh, GCC codes for alanine. This keeps occurring, so once another amino acid has attached to the top, this keeps occurring. UGG is trypsin. ACU will be threonine until the entire mRNA sequence has been translated into amino acids. And again, amino acids are the building blocks of proteins. So you're forming an amino acid chain. An amino acid chain is just a long strand of amino acids attached together. The end result of translation is an amino acid chain, and that folds up into proteins. So looking at a slightly different picture, we still have the mRNA here, we still have our ribosome here, and each of these little circles attached along the top is an amino acid, so a string of amino acids. The proteins in humans are made up of 20 different kinds of amino acids. And again, 3 nucleotides will give you 1 codon, and each codon codes for 1 amino acid. Now that we've talked about replication, transcription, translation, let's have a quick summary of each one. DNA replication, remember, the end result is 1 DNA turns to 2. This happens in the nucleus. Transcription, DNA turns into mRNA, also in the nucleus. And the new process we learned, translation, is when mRNA turns into amino acid chains, which become proteins. And this happens in ribosomes. Try these 3 questions. What happens in transcription? Where does it happen? And what happens in translation? Make sure for the first 2, you're answering what happens in transcription, and the last one's about translation. Transcription is mRNA is made from DNA. It happens in the nucleus. And translation is mRNA, which was made during transcription, is turned into amino acid chains, which eventually make proteins. Next, try these 3 questions. Which of these processes does not use DNA? Well, translation turns mRNA into proteins. No DNA is involved. Transcription is when mRNA is made, and translation is when mRNA is used and tRNA attaches on and eventually makes amino acid chains. Alright, now stop the movie here and go back and watch any parts you don't understand so that you fully understand what translation is. Then answer a question about translation before continuing on with the video. The next question you have to ask yourself is, how do you figure out what amino acid is going to attach on? To do that, use something called a codon wheel. Now, using a codon wheel is pretty simple, even though it looks complicated to start. Let's say you were given the codon AUG. The first letter of this codon is A. So to use a codon wheel, you go to this picture over here, which you would be given, and since it's the first letter, you go to the biggest inner circle and find the letter A. You can see that A is located right here. So now, nothing in the rest of this codon wheel matters if it's not in the piece of the circle with the A. So you can pretty much get rid of all this and just concentrate your attention on that A quarter of the wheel. Now the next letter is U. So now you look for the medium letter with a U, and you find it right here And so you know that none of this part of the wheel is going to matter, but you're only going to be looking in this segment here. When you zoom in on the U segment, the last letter you're looking for is G. So you go to the smallest outer circle, you find G, and you read that the amino acid that AUG codes for is called methionine. A lot of times that's abbreviated just MET, or meth. Try these two practice problems. What is the amino acid for UUC? And what is the amino acid for GUG? Use this codon wheel to help yourself find the answers. The first question asks, what is the amino acid for UUC? To figure out the amino acid for UUC, again, you're going to find the U in the biggest circle. So you're only looking in this section. Next, you move to the next U, here. So now you're isolating where you're looking to this section. And finally, C here gives you phenylalanine, which is a lot of times abbreviated PHY. The second question asks for GUG. Use this codon wheel to find GUG. Again, you want to start with the biggest section and isolate it so that you know you're just looking here. Next, you go to the medium letter, find the U, and know that you're just looking here. Finally, you find the G, which is located right here, and say the amino acid is valine, or val. Try these 3 check yourself questions. Given the RNA sequences, what is the protein sequence? Remember, to start you need to break up the RNA sequences into codons, and then go back and use the codon wheel from the video. It might help to write these on a piece of paper so that you don't have to just have it in your iPad. If you need help with the first check yourself problem, here's how you would break it down by step. Step 1 is to break up the sequence into codons. So this sequence here has simply been broken up into codons, or groups of 3 letters down there. Step 2, look back for each codon in the codon wheel. And what you find is AUG codes for methionine, GGU will give you glycine, CUG will give you leucine, and AUC will give you isoleucine. Make sure this is the results you got for the first question. Then go back and try all the questions. At this point, you finish the video entirely. You should know what translation is, where it happens, what it does, and also be able to see an RNA sequence and translate it yourself into a protein sequence of amino acids. If you can't do any of these things, go back and watch the part of the video you need.